Thank you for joining us, and welcome to Kaiser's webinar, Raman Spectroscopy with Synchrotron X-ray-based techniques, a powerful approach in materials science and heterogeneous catalysis. My name is Lisa Ganster, and I will be your webinar host. Today's guest speaker is Dr. Valerie Briwa, Beamline Manager of the Rock Beamline at Scie Synchrotron. During the webinar, Dr. Briwa will discuss how Raman and Synchrotron X-ray-based techniques have been combined at Scie's Samba Beamline. In addition, she will illustrate the power of the complementary techniques using selected examples. I'd now like to introduce Dr. Valerie Briwa.
Hi, everybody. Thank you very much, Lisa, for this introduction. So, the purpose of this webinar is to show you how complementary and powerful is the combination of Raman spectroscopy with X-ray absorption spectroscopy. Here, you have the outline of my presentation. After a short introduction to X-ray absorption spectroscopy, I will discuss the common specificities of both techniques, and after that I will present you how this combination of technique can be carried out. And finally, I will illustrate with two examples belonging to my own research the complementarity of these techniques in the field of sol-gel chemistry and in the field of heterogeneous catalysis. Let me start with the X-ray absorption spectroscopy. The X-ray absorption spectroscopy refers to the measurement of this absorption coefficient here, mu, as a function of the energy of photons here. And this absorption coefficient is related to the intensity of the incident beam, E naught, and to the intensity of the transmitted beam, I, through the well-known Beer-Lambert relation. Here you have a typical X-ray absorption spectrum recorded for nanoparticles of cobalt oxide, and this has been recorded at the cobalt K edge. You can see in this plot several regions of interest. When the energy of photons here is equal to the binding energy of the 1s electron of the cobalt, you have this abrupt increase of the absorption coefficient, which is called the edge. Then, at the top of this edge, you have this main resonance and several resonances just around. This part is the so-called Xense spectrum. Xense is the acronym for X-ray absorption near edge structure. Far above the edge, about 50 to 100 electron volt above the edge, we have the region which is the EXAF spectrum, which is called the EXAF spectrum. EXAF is the acronym for Extended X-ray Absorption Fine Structures. The EXAF technique is a well-known local order technique, and a figure of merit of this technique is the well-known Fourier transform of these EXAF oscillations. Here you have the Fourier transform of the EXAF oscillation for this compound, for this spectrum. The Fourier transform is a pseudo-radial distribution function of neighbors around the absorbing atom. In the case of this cobalt oxide rock salt structure, we have as first coordination shell, we have oxygen neighbors, which are located at 211 angstrom, and this first intense peak here is related to this first coordination shell. At longer distance, we have the second nearest neighbors, the cobalt second neighbors, located at 3.02 angstrom. And as you can see here, we have also signal at longer distances, but you see that this signal is very small in intensity. This is due to the fact that the EXAF technique is a local order technique only sensitive to neighbors located up to, let's say, 5 to 6 angstrom far from the absorbing atom. The analysis of this EXAF oscillation and of the Fourier transform will give you as parameters, as structural parameters, the coordination numbers around the absorbing atoms, the nature of these neighbors, I mean oxygen, cobalt, or what you want, and the distances between the absorbing atom and the neighbors. Now, let me give you some words about the Xen's part. The position of this receiving edge here, that means the position of the 1s electron, is very sensitive to the oxidation states, and as you can see here with this plot, you have a shift of the edge position when you increase the oxidation state of the absorbing atoms. The black curve is related to a cobalt foil, where you have as oxidation state cobalt at more reduced state, cobalt 0. Then the blue curve, the blue spectrum, is related to the cobalt oxide, where you have divalent cobalt atom, and the red one is related to the spinel-like oxide, where you have a mixture between cobalt 2 and cobalt 3. And you see that we have a clear shift of the position of the receiving edge with the oxidation state of the absorbing atoms. That will be an information very important that we will use later on the seminar. There is another point which is important related to the Xen's part, is that, as you can see here, the intensity of the main resonances here is also strongly dependent on the oxidation state of the absorbing atom. We have a more intense contribution when you have oxidic species compared to the more reduced state. That's more or less all you need to know to follow me in this seminar regarding the X-ray absorption spectroscopy. Let me finally summarize some of the specificities of the X-ray absorption spectroscopy. This technique is atomic selective. This technique can be applied to all kinds of materials, and that over a very wide range of concentration. And we can use the X-ray absorption technique for in situ characterization of materials. That's our important points that will be used later on this webinar. The only drawback, let's say, of the X-ray absorption spectroscopy is that this technique is not a laboratory technique. This technique requires an intense X-ray source to be applied, such as the source provided by a synchrotron radiation facility. Here you have a view of the synchrotron radiation facility located near Paris in France, the so-called Soleil synchrotron radiation facility. And all the results that I will present you today has been recorded using the so-called SAMBA beam light, as you can see here in this picture. SAMBA being the acronym for spectroscopies applied to materials based on absorption. Now, dealing with the combination of synchrotron radiation-based techniques with Raman. First, Raman. Raman is a well-known laboratory local order technique which provides rotational and vibrational information which is specific of a chemical bond. And the combination of Raman spectroscopy with a synchrotron radiation technique is not really new, because if you look at the research done by colleagues in the past, the first researcher who combine synchrotron radiation technique like the small angle X-ray scattering or the wide angle X-ray scattering with Raman was Wim Brass in more or less 20 years ago for the study of polymers. Now, about 10 years ago, my group was among the first group combining X-ray absorption spectroscopy with Raman spectroscopy. And more or less concomitantly, colleagues from Brookhaven, I guess, and colleagues from the European Synchrotron Radiation Source combine X-ray diffraction technique, the X-ray diffraction technique, with Raman spectroscopy. Considering the specificities of X-ray absorption spectroscopy that I previously described, with the advantages of Raman, which can be found, for instance, on the Kaiser website, you see that the combination of these techniques appears quite as a natural union, because these two techniques, both techniques, share a lot of advantages, a lot of specificities. These specificities are, of course, the atomic and bond specificity. These two techniques can be applied to all kinds of materials over a very wide concentration range. And on top of that, these two techniques can be used in order to characterize dynamical processes with a very high time framing. The combination of Raman spectroscopy with X-ray absorption spectroscopy was only made possible thanks to the development of a new generation of compact spectrometers based on optical fibers. Here you have a view of the setup used, for instance, on my beamline. This new generation of spectrometers incorporate a laser diode as excitation light and an efficient detector for the Raman signal detection. The laser here is incident on the sample through the Raman probe head. This Raman probe head hosts a single optical fiber for the laser excitation and another one here for the collection of the scattered Raman signals. The Raman probe head, like this one, here you have some accessories which can be found on the with the Kaiser corporation. This Raman probe head can be equipped with different objectives. If you want to study solids, you can use non-contact objective like here. If you want to characterize a liquid solution, you can equip the Raman probe head with an immersion probe. And more recently, you have the possibility to study gases using a specially designed Raman probe head for gas. Actually, the versatility of the optics that you can mount on this Raman probe head make very comfortable the combination of Raman with X-ray absorption spectroscopy. You have only to take care on the X-ray absorption constraints in terms of let's say thickness of the sample, of optical windows, and most of the time the Raman spectroscopy is able to manage the constraints of the X-ray absorption spectroscopy. So, at Soleil, on my beamline, on the Samba beamline, we have two COSY Raman RXN1 spectrometers. One of these spectrometers here is equipped with a near-infrared laser diode working at 785 nanometer, and we have another one equipped with a green visible laser diode working at 532 nanometer. Okay. Let me start with the first example. This example deals with the in-depth study of the genesis of tin oxide nanoparticles. This study was performed in collaboration with Brazilian colleagues and was recently accepted for publication, as you can see here, in Chemistry of Materials. The purpose of this study was to obtain a fine and complete understanding of the mechanism involved during the formation of tin oxide nanoparticles, and that in order to control in a predictable way the size of the nanoparticles. Indeed, it is well known that tin oxide nanoparticles have a high technological importance in many fields, these fields being listed here. But efficient tin oxide nanoparticles can be only obtained through a careful control of the size, morphology, and degree of aggregation of the particles. And in order to improve our knowledge of mechanism leading to the formation of these tin oxide nanoparticles, we carried out a complementary structural characterization using complementary technique with a very high time framing. This technique was the small angle X-ray scattering, the X-ray absorption spectroscopy, and of course, we combine the X-ray absorption spectroscopy with Raman spectroscopy. So, the preparation route for the preparation of tin oxide nanoparticles is very easy. This route is based on the slow addition of an excess of water into a solution, an ethanolic solution of tin tetrachloride pentahydrate. And we studied the formation of these tin oxide nanoparticles during the addition of water, which took more or less 10 minutes, and the further aging at room temperature. So here you have the setup used for the in-situ monitoring of the hydrolysis of the solution and subsequent aging. And you can see here that the Raman spectra were collected using this Raman immersion probe, which is in direct contact with the solution through a special aperture here at the top of the cell. The data for the X-ray absorption spectroscopy were obtained using the same cell, of course, and in transmission modes. That means that we let the X-rays to pass through the cells using transparent X-ray windows. The typical time resolution for the collection of Raman was 1 spectrum every 5 seconds, whereas for the quick EXAFS, means for the time resolve X-ray absorption data, we could collect 1 spectrum every 500 milliseconds. But in order to improve a little bit the signal-to-noise ratio, we merge 50 spectra, and the real-time resolution actually is 50 seconds for the X-ray absorption spectroscopy. So here you have the results obtained during the addition of water and aging at room temperature and gained by X-ray absorption spectroscopy recorded at the tin KH. That means that we are just looking at the local order around tin. Here in this Fourier transform, you have an intense contribution here, and this intense contribution here marked by this green arrow is related to the chlorine ligands, chlorine atoms in the first coordination shell around tin. The shoulder here, marked by the red arrow, is related to the oxygen atoms in the first coordination shell around tin. So it is quite obvious that we have, during the addition of water, a decrease of the contribution related to the chlorine ligands and a small increase of the contribution related to the oxygen atoms. A more quantitative results can be obtained through a least square fitting of the EXAFS coordination number by looking at the least square fitting of the EXAFS data. Here you have this EXAFS coordination number determined for chlorine and for oxygen during the water addition and aging. And you see that we have, during the water addition, a change of the coordination number passing for chlorine from 5 to about 4, and an increase of the coordination number for oxygen passing from 1 to 2. This results from the exchange of chlorine ligands by water molecules, during the addition of water. Here you have the results collected at the same time by Raman. I just report the data related, the interesting part of the data, which is related to the stretching vibration mode for tin chloride bond. And as you can see here, we have results which are in agreement with the X-ray absorption results, because you see that upon addition of water, we have a decrease of the intensity of the main contribution here, which is related to this exchange of chlorine by water molecules. We can have more quantitative insight on this combination just by comparing the intensity here of the bond area, of this bond area, as a function of time. It deals with the blue curve here, which is compared to the coordination number for chlorine atoms. And you see that both curves are totally superimposable, showing that the combination, that this kind of combination of technique is well adapted. Nevertheless, at this stage of this combination, we have only a global information about the species involved during the addition of water and during the formation of nanoparticles, because we have only an idea of the number of chlorine atoms and of oxygen atoms surrounding the tin absorbing atoms, but we have no real identification of the species. If you look at the literature, you can see that this broad band here is the overlap of several lines, each line being characteristic of a kind of complex, of a kind of allied tin hydrated complexes. Here you have the results that you can find in this publication, for instance. When you decrease here the number of chlorine ligands around tin, you have a redshift of the position of this line, as you can see in this curve. Using this information, I mean the well-known fingerprint for each complex, and using a deconvolution procedure of the Raman data collected at any stage of the reaction, you can achieve a very good description of the composition of the solution. For instance, in the case of the data recorded before water addition, you have this deconvolution, which gives as a result about the same quantity proportion of pentachloride and tetrachloride, and only 18% of trichloride. So, considering this peak deconvolution for all the data recorded by Raman during the water addition, I mean during the first minutes of the reaction and further during the room temperature aging, deconvoluting this data, we obtain this tin allied speciation, where you see that during the water addition we have a decrease of the proportion of the more of the complexes containing the higher amount of chlorine atoms, the pentachloride and the tetrachloride in blue and red respectively, and this at the benefit of the formation of the more hydrated trichloride species. That are the results gained by Raman spectroscopy. Of course, we have at the same time the results gained by X-ray absorption spectroscopy, and this is presented here and illustrated here in terms of quantitative speciation. We know from small angle X-ray scattering in particular that we have the formation of tin oxide particles at the early stage of the water addition. And taking into account this information, we consider for any X-ray spectrum recorded during the water addition and recording during the room temperature aging, we consider that we can rebuild the experimental data, which is here represented in blue, by a linear combination considering tin oxide nanoparticles and the signal for a precursor solution. Here you have the components used for the rebuild of the experimental data recorded at the end of the room temperature aging. So you see that we have a very good agreement between the linear combination and the experiment. That can be done in the K space, that means on the exafosylation, but also in the Fourier transform. Using this linear combination fitting here, you can describe the change in concentration, in proportion of tin oxide nanoparticles during the full time of the reaction. So, now, let me combine all the results gained by Raman spectroscopy here with the colored samples and with X-ray absorption spectroscopy gain with the linear combination of the exact signal for the tin oxide nanoparticles. This analysis of tin speciation provided by both techniques was really decisive to propose reactional pathways leading to the formation of tin oxide and to make links between the different species here represented here, which was put in evidence by small angle X-ray scattering. We know that from small angle X-ray scattering that we have in presence during the reaction, first monomeric species, later on we have these linear chains made of edge-sharing octahedra, and at the end, of course, we have these tin oxide nanoparticles. So, taking into account the speciation here, we were able to propose the following reaction pathway. First, at the early stage of the reaction, that means before, during the addition of water, but before the nucleation. The nucleation is the time where you have the formation, you start to form the tin oxide nuclei. So in this region, we evidence a covariation of the penta and tetrachloride complexes with the trichloride complexes. And as a matter of fact, as you can see here clearly, we have the rate of formation of this trichloride, which is twice the rate of consumption of the two other complexes. That means that the equilibria, the reaction which are responsible for this change of concentration are this one. At the advanced stage of water addition, during the nucleation stage, we saw that this related to this species, the trichloride species, we saw a plateau. That means that this species is not responsible for the formation of tin oxide nanoparticles. Actually, the consumption here of the tetrachloride complex, the red one, appears more marked than the consumption of the pentachloride, and this is due to the fact that this tetrachloride complex here is involved into olation reaction between monomers in order to form these chains obtained by edge-sharing octahedra. And finally, during the room temperature aging, the chains with this edge-sharing octahedra and well evidenced by small angle X-ray scattering are involved into a cluster-cluster aggregation in order to form these tin oxide nanoparticles. So, dealing with this first example, I would just to mention that without this complementary information gained by EXAF and Raman spectroscopy about the species in solution, we were not able to describe so completely the mechanism leading to the formation of these tin oxide nanoparticles. So, let me move to my second example. This example deals with the study of heterogeneous catalysts in working condition. The studied catalysts are cobalt-based catalysts used for the Fischer-Tropsch reaction. The Fischer-Tropsch reaction is a reaction which is carried out at quite high pressure, between 20 to 30 bar, and at moderate temperature range between 230 and 280 degrees Celsius. This reaction converts the syngas, I mean the carbon monoxide and the hydrogen, into liquid hydrocarbons and water. The scope, of course, is to form these liquid hydrocarbons in order to have, at the end, these liquid synthetic fuels. The active species for the Fischer-Tropsch reaction are metallic cobalt nanoparticles, which are dispersed at the surface of an alumina support. In this combination of technique, we try to improve our knowledge of the activation process of the catalyst, partly. And to do that, we need to have an operational approach. That means to record simultaneously information about the catalyst state during the reaction and the catalytic activity of the system. And to do that, we used Raman spectroscopy in order to assess this catalytic activity. And of course, as you will see later, we use X-ray absorption spectroscopy in order to have information about the catalyst state, the state of cobalt at the surface of alumina. So, in this second example, I use a lot the Raman probe head suitable for the analysis of gases. This Raman probe head that you can see here can be directly mounted into the gas slipstream using Scie rock fitting. And this Raman probe head was used in order to monitor the partial pressure in gaseous reactants, that means in hydrogen and carbon monoxide. Okay, so here you have the typical Raman spectrum, which can be recorded as the catalyst is under syngas at 20 bar. We have first well-defined lines here, as you can see, here and here, which are related to the gaseous reactants, the hydrogen and carbon monoxide. And we have this region here, which is centered around the region of CH stretching mode, where you have information about the reaction products, the hydrocarbons formed by the reaction. The first stage, the first step that we have to do in order to quantitatively monitor the catalytic activity for this reaction, of course, was to record calibration curves for both gas size. So it's very easy to do that by increasing the pressure of reactants, of gases inside the cell. We have a linear dependency of the intensity of the line as a function of the gas pressure. And using this kind of monitoring of Raman intensity as a function of the pressure, you can access to this calibration curve. So that was the results obtained for one of our catalysts. Here you have, as a function of time under gas stream, you have the variation of the peak area for the hydrogen and for the carbon monoxide. These data were recorded at the outlet of the cell. Here you have the cell in which you have loaded the catalyst, and just at the outlet of the cell we install this Raman probe head for the analysis of gases. So using this information and the previously described calibration curve, we can determine the partial pressure in carbon monoxide at the outlet of the cell, and dealing with the partial pressure at the inlet of the cell. It's very simple because we know the pressure inside the full line. So we know the ratio between hydrogen and carbon monoxide, and so we can determine pretty well the carbon monoxide conversion rate. Higher is this carbon monoxide conversion rate, more active is the catalyst, of course. Okay. Now, let me consider the results recorded by Raman over 20 hours of reaction for one of our catalysts. Here you have the intensity, the relative area of the different products analyzed by products and reactants analyzed by Raman spectroscopy. You have in blue the hydrogen, you have in pink the carbon monoxide, the gaseous reactants, and you have in black and green the products of reaction. And that is recorded over 24 hours with some changes in temperature, since we pass from a very high temperature of operation for the catalyst, 270 degrees Celsius, to a lower temperature, 230 degrees Celsius. And using the calibration curves previously described, and using these results, we were able to quantitatively describe as carbon monoxide conversion, a very high conversion ranging of about 76% for the high temperature to 8% for the low temperature. That means, as previously, or as you can see here clearly, seen from this variation of intensity for the different reaction products, the catalyst is more active here at high temperature than at low temperature. Now, that's for the catalytic activity. Now we can use as well the Raman spectroscopy in order to have information about the selectivity of the catalyst, of the reaction towards the different products. The Fischer-Tropsch reaction is comparable with the polymerization process. That means that we have at the end of the reaction a distribution of products with variable chain lengths. And as you can see here, with the comparison of the Raman spectra recorded for pure references, I mean for methane, ethane, propane, and for hydrocarbon with longer chains, we have in this data recorded for the products of reaction at the different temperature, we have an overlap. We have a distribution of reaction products. At very high temperature, we see that we have this intense line here, which is a characteristic of the formation of methane. But when we decrease the temperature from this to this, we observe a huge decrease of this main line characteristic to the methane. That means that we form at lower temperature hydrocarbon with longer chains. That's for the qualitative description, but we can go beyond this qualitative description by using chemometric tools. And now, using on the same set of data, the CH stretching vibration mode recorded at the different temperature for the products of reaction, we can use this set of data and the so-called multivariate curve regression alternatively square fitting procedure in order to determine the concentration, the proportion of the different components. And in this case, we see clearly that at high temperature, I mean at this temperature, where we have the more active catalyst with about 76% of conversion, we form mainly methane at a level of 68%, and only 11% of products is related to alkane with long chains. Now, if you look at the result obtained at low temperature, for the less active catalyst, we have the reverse results. We have a very high amount of alkane with long chains, about 66%, and only 30% of methane. That means that despite the fact that this catalyst is less active, this catalyst is more selective towards the formation of the products of interest for the Fischer-Tropsch reaction. That means towards the formation of long chains products. So that's for the results gained by Raman dealing with the catalytic activity and dealing with the specificity of the catalyst at the different temperature. Now I would like to discuss these results in conjunction of the X-ray absorption results gained simultaneously to the characterization by Raman. Here you have, for instance, the Xen's spectra recorded at high temperature. These spectra recorded at 270 degrees Celsius, where you have this carbon monoxide conversion. If you look at the maximum of the white line here as a function of the residence time at this temperature, we observe that during this residence time at high temperature, we have an overall increase of the intensity of the white line, as you can see here with the zoom. That means that during the high temperature reaction, we have a slight oxidation of the catalyst, and this can be pretty well understood if you consider the catalytic activity. Indeed, if you have this very huge conversion of this carbon monoxide and hydrogen, that means that you have a huge formation of water. And regarding this huge formation of water, the system reacts in order to shift this reaction towards the left, and that means to change the active species towards a less reduced species, towards this cobalt oxide species. That's for the results obtained at a given temperature. Now, if we compare the different results gained by Xens at the different temperatures characterized during 20 hours, we see that by decreasing the temperature from this 270 degrees Celsius to 230 degrees Celsius, we have a decrease of the main intensity of the white line. That means that we have a slight reduction of the oxidic species towards metallic species. And again, this can be well understood considering the carbon monoxide conversion, because we decrease the activity of the catalyst. That means that we decrease the formation of water, and in this case, the remaining cobalt oxide species can be slightly reduced into active cobalt species. So, yes, it's the end of the second example. I hope that you are convinced with this second example dealing with heterogeneous catalysis that we can provide very complementary information related to the catalytic reaction, the catalytic activity and the selectivity using the Raman spectroscopy, and the catalyst state, a discussion related to the radius versus oxidic states using the X-ray absorption spectroscopy. So let me finally make a general conclusion about this combination of Raman spectroscopy with X-ray absorption spectroscopy. I hope that you are convinced that such a combination is very powerful for the study of dynamical processes through the complementary information that you can obtain with the two techniques. But our approach, at least in my mind, was clearly beyond this complementarity of information, because, for instance, with the genesis of nanoparticles, of tin oxide nanoparticles, we were able to determine accurately the order of occurrence of the different events, of the different reactions, with a very high time framing, and this can be only done if you have combined experiment and not separate experiments. This is the only way to have a non-ambiguous interpretation of the results. In a second point, I would like to say that the combination of of two techniques, Raman and X-ray absorption spectroscopy, but anyway of other techniques, can be very important in order to eliminate artifacts introduced by separate experiments. And these artifacts could be related to the history of the sample, to the sample environment, to the temperature change, what you want. finally, as the last point, considering that we are dealing with techniques where you have an interaction of electromagnetic radiation with samples, the use of an additional techniques of characterization can be also very helpful in order to detect sample damage. And this is particularly true for the use of X-rays produced by a third-generation machine. We have a very high flux, and most of the time the samples suffer from this high flux of photons. So for very sensitive samples, such a cross-check of, let's say, good health of the sample can be crucial. Well, I think that, yes, I am at the end of my talk. I would like to thank all my collaborators who contribute to the results that I presented in this webinar, and thank you for your attention.
Thank you, Dr. Briwa. And as we mentioned, we will take a few moments to answer any questions that you've submitted.
Okay, so we have one question coming from Dr. Fisher. The question is, I know that the question is related to the Fischer-Tropsch reaction. And the question is, are these selectivity trends confirmed via chromatographic method? The answer is, we confirm the selectivity trend not by chromatography method, but by mass spectrometry, and we saw really that at high temperature we have the formation of more methane than the other long chain hydrocarbons, and the reverse at low temperature. Okay, thanks. One more. What sizes were the cobalt crystallites? The typical sizes for the cobalt crystallites was about 10 nanometers. If I have the good value in mind, it's not so very small crystallite size. It's more or less comparable to the size that you have in the oxidic form at the beginning. About 10 nanometers.
We'd like to thank everyone for attending, and a special thanks to Dr. Valerie Briwa for presenting today. If you have additional questions after the session today, please feel free to send them to marketing@kosi.com, and you may register for future webinars on the Kaiser website or request archive sessions at www.kosi.com. Thank you for joining us today, and we hope to see you at the next session.